How are you? Yeah, with a yeah. They show me what the claims to me, I think it's that. Yeah, so you have
Yep.
So, yeah.
Muy bien, bienvenidos todos de nuevo. Los invitamos a que tomen sus asientos. Voy a comenzar con las instrucciones de seguridad. En caso de que suene cualquier alerta, la mitad del auditorio tiene que evacuar por esta puerta y la otra mitad por la otra puerta, es decir, la que les quede más cerca, y después reunirse en el punto de reunión que está en el estacionamiento marcado con verde. Les recuerdo que no pueden consumir ningún alimento ni bebida aquí dentro del auditorio. Y también, pues, si de una vez pueden sacar sus celulares para ponerlos en silencio o apagarlos. Y finalmente, quiero recordarles que gracias al financiamiento del posgrado en Ciencias Biológicas y también al financiamiento de la Coordinación de la Investigación Científica es que podemos tener estos seminarios tan interesantes para nuestro festejo del 95 aniversario. Sin más, voy a dar paso al doctor Gerardo Salazar, quien va a presentar a nuestro ponente.
Buenos días a todos. Bienvenido, Oscar. Otros invitados. Welcome to Mexico. Thank you for being with us. Tengo el gran placer y orgullo de presentar al doctor Oscar Alejandro Pérez Escobar, quien es un biólogo evolucionista y además taxónomo de plantas, cuyo interés de investigación principal es entender los factores que subyacen aspectos microevolutivos y macroevolutivos de muchos grupos de plantas, pero con una pasión muy definida por las orquídeas. Es un hombre de muy buen gusto. Oscar obtuvo un grado de ingeniero agrónomo en su nativa Colombia y posteriormente el doctorado en la Universidad Ludwig Maximilians en Múnich, Alemania. Y actualmente, ya desde hace varios años, ocupa una posición permanente como líder de grupo de investigación y es beneficiario de la Sainsbury Orchid Fellowship en los Reales Jardines Botánicos del Reino Unido. Oscar tiene una... el Reino Unido, disculpen. Oscar trabaja con una enorme comunidad internacional. Si ustedes ven la lista de coautores en sus publicaciones, es extensa, tiene muchísimos amigos. Y pues él estudia los patrones de riqueza de las especies vegetales, así como sus patrones de distribución, de especiación, extinción, básicamente utilizando datos de magnitud genómica que él genera y además analiza. Tiene 86 publicaciones en revistas de muy alto prestigio, incluyendo Nature, Science, PNAS, etcétera. Y déjenme comentarles que algunos de sus trabajos más leídos incluyen la secuenciación de una sandía de 6,000 años de antigüedad, el que ya se comentó de una palma de 3,000 años, analizando los genomas, pero también tiene muchos otros trabajos súper importantes, integrativos, sobre el origen de la diversidad y la evolución, por ejemplo, de la flora andina y entre muchos otros de la gran diversificación vegetal que representan las orquídeas actuales. Entonces, tengo el placer de presentarles a mi estimado amigo Óscar Alejandro Pérez Escobar.
Bueno, antes de comenzar, quisiera en verdad agradecer al Instituto de Biología y a los organizadores de este simposio por la invitación. De verdad que es una gran felicidad y orgullo poder estar compartiendo aquí con amigos y colegas, así que muchas gracias. Well, I guess as some of you know, most of the research I have been doing so far is centered in the, well, we could say one of the most beautiful and wonderful plant families, flowering plant families on Earth. That remark goes with apologies to my fellow legume experts and other monocot experts. But today, sorry? I had a colleague once that said, and as a Pérez expert, that orchids are overrated. That sometimes I tend to agree with him. Sorry, Gerard. But then I thought about today breaking a bit of protocol and go beyond what I'm being paid for in Q, which is the orchid research. And I thought about sharing with you a research project I've been working with my collaborators and I, which is very close to our hearts, and is about the origin and evolution of what we think is the world's most misunderstood plant. And that is the coca tree, right? So this is something we started somewhere 4 or 5 years ago as a side project, but has been growing, as you will see. We like to say that the coca tree is one of the world's most misunderstood trees, because the first thing that comes to mind when you talk to colleagues and the general population about this important plant is that coca equals cocaine. So when I say cocaine, I don't speak about the tropane alkaloid itself, but the recreational drug that people consume in parties and stuff. But I can tell you that coca goes way, way beyond a drug. And as a matter of fact, the cultivated cocas belong to a genus that we call Erythroxylum, which from Latin means redwood. And is a huge genus that has over 270 species that occur in the pantropical regions of the world, but with 50% of the diversity that is found in the American tropics. Of all the species that are known to, that have been formally described, 2 are cultivated, actively cultivated in a traditional and in an illegal way, as we will see later. And these are basically Erythroxylum coca and Erythroxylum nomogranatense. So each of these 2 taxons have 2 varieties, Erythroxylum coca, variety coca, Erythroxylum coca, variety padu, and so nomogranatense, right? And what we know so far is that each of these 2 species have 2 close wild relatives, which are Erythroxylum cataractarum and gracilipes. So we know this thanks to the pioneering research that Dawson White and collaborators conducted some years ago. What you can see in this phylogeny, which was actually built using 400 low copy nuclear genes and population level sampling, is that in this group here of all mostly neotropical taxa here, you have Erythroxylum nomogranatense and coca nested in the wild relative gracilipes, and somewhere here is cataractarum. So this led to Dawson White collaborators subsequently to propose a new domestication scenario of this crop, where basically these nested populations of cultivated cocas that you see here, here, and here were derived from the wild ancestor gracilipes, right? But we don't really know where this pattern that we are seeing here, how much subject is from hybridization, gene flow, and so on and so forth. So what you see here is the distribution of the cultivated cocas and the wild relatives. And of all the 270 species, as far as we know, the ones that we have analyzed by means of GCMS, so that's gas chromatography and mass spectrometry, that only Erythroxylum coca nomogranatense produce copious amounts of cocaine, right? This is the distributions of the cultivated cocas. So these purple, pale purple circles and green shades, and then the other areas are like the wild relatives and outgroups. So how do we know that they are like wild relatives don't produce cocaine? And this is basically thanks to analysis that we have been doing before and as this ongoing research, where we have analyzed samples from our historical collection. And what you see here is that when you piled up all the gas chromatography analysis, you can see here a huge peak, which basically codes for cocaine. But when you compare the same profiles for other samples of the wild relatives, that peak is absent, right? These analyses were done by my colleague, Federico Padilla, at that time when he was working in Kew Gardens. But then coca goes beyond what I was saying before, cocaine. And I think that one of the less known aspects of the coca tree is the tradition that it has had for actually thousands of years. So we have microfossil records, or what I like to call archaeobotanical records associated to human use, which go back to 8,000 years, found in dry areas of Peru and Chile. But one of my favorite items is this. This is basically a moche sculpture, which was found somewhere here in what archaeologists call the Moche Norte region, northern region. And what this sculpture depicts is apparently a man which is holding an item on his right hand and another item on the left hand. And this actually can translate to a tradition that survives still today. So this is the act of chewing coca leaves using items that we will see before, right? So this sculpture actually dates back to 1,200 years ago. And so the importance of the coca tree is vast and reflects the importance it had for humankind, like for South American people until present. So it's not only one scattered records, actually hundreds of records. These are another favorite of mine. This is what we call a Colombian poporo, which is an alloy of copper and gold, and is 1,700 years old. We have tupu pins which were worn by different indigenous communities and were used to were associated to the use of chewing coca leaves, right? So this is not just isolated records. We have ample evidence that coca leaves have been used by humans for different purposes, for the medicinal purposes, as a food supplement, as a stimulant, right? Nothing, again, to do with the, getting high and, just using cocaine for fun. And I said before that this item is one of my favorites, because what you see here that was depicted 1,200 years ago is being done exactly the same way today, which is mind-blowing. This person that you see here is called Ismael. He's a mamo, which I guess in English it would translate into a shaman. And what he's doing here is poporear. So this item that he's holding here is the same item that this sculpture is holding. It's a gourd. It's an item made of a gourd where calcareous powder is deposited in. There is a wooden stick, which supposedly is what this person was like holding in its hand. And then what Ismael is doing is that he's taking coca leaves from his back, which are dried, putting a mouthful in his mouth, and then chewing it. And as he chews, he goes with a stick and takes out the calcareous powder and put it in his mouth. So the idea of the calcareous powder is basically helping drawing the alkaloids from the leaves, right? The calcareous powder is sourced from different ways. Traditionally, there is a tradition that says that indigenous people, for example, in Sierra de Santa Marta in northern Colombia, used to go down the mountain in a pilgrimage, collect seashells, burn them, and then bring that calcareous powder and mix it with the leaves. So again, this is a tradition that survives today and tells the importance of it. And the same traditional use reflects in a diversity of languages and the name of the plant has in different communities. For example, for this Arhuaco male, in his native language, Ika, they call it Haju, the Huitotos call it Hibie, the Quechuas call it Cuca, and Tucano call it Patuca. So I think that Michael was saying before about the importance of traditional indigenous names or traditional names, and that reflects the importance of a plant in communities. So this is still very much alive today. So if some of you have wondered how indigenous people grow coca leaves or coca trees, that's the way. It has nothing to do with industrial scale plantations that drug barons have established in different countries, like mine, for example. So it's a traditional chakra where you have coca bushes here, and it's associated with many crops like corn, tobacco, manioc, sugarcane in the background, banana trees. so on and so forth, right? So this is one side of the coin, the traditional use that has existed for thousands of years. And then there is sadly the more recent illegal use, which is extensive plantations of coca trees for the synthesis of cocaine and the production of the cocaine drug itself. What you see here in these photographs, which was taken by a colleague of mine, Jose Aguilar Cano, is the aftermath of a population that was abandoned, which was basically made in the center of Amazonian forest in Colombia. After the declaration of war on drugs by President Nixon in 1971, this is like the scenario in countries like mine and Ecuador, Bolivia and Peru, where basically has resulted in the displacement of about 7 million people on the last 50 years and the murder of the death of hundreds of thousands of people, right? So these numbers are not directly linked to the cultivation, illegal cultivation of coca plantations, but of all the violence that is associated with it and illegal groups, of course. And this is, of course, a non-trivial matter for biodiversity as well. What you see here is a plot that shows the persistence of populations, illegal coca plantations in the country, and in red shades and yellow shades are the ones that have been persisting the most. And as you can see, some of them are actually nested in important biogeographical regions, biodiversity hotspots like Northern Andes. So if you overlap this map of Colombia somewhere here, you can actually see that some of the areas, some of these plantations are nested in like one of the most biodiverse places on Earth, right? Which we keep deforesting and expanding. So we are also losing biodiversity with the coca plantation. But if we then translate into what is like crop domestication or genomics or evolution, there is a really fascinating aspect. And it's that all these illegal plantation things have apparently led to the unsupervised breeding of like what we call super varieties. So if you read here this headline from a newspaper from 2021, narcos alteran la coca para hacerla gigante y persistente, right? So even talks about like genetic manipulation. Like, so it seems that like drug barons have been investing some good money in agronomists, trying to improve varieties to survive, you know, what these aerial dispersants with herbicides and produce more alkaloid, or creating, for example, gigantic plants that produce more leaf and then therefore increase the yield of leaf and therefore of cocaine per hectare. So from someone that has interest in crops, that's like mind-blowing, really. Like the research opportunities are fascinating. I'm just trying to get something positive out of it, right? And because of all, you know, the illegality of the plantations and the social problems, there have been a lot of funding investing on monitoring how these plantations have been growing and what have we been growing in these plantations. And so the United Nations Drug and Crime Office have been doing monitoring for about 20 years on how these populations are distributed in Colombia and different countries. And here you see an example of one of the reports that's from 2021, which basically says that production of cocaine has, increased, and the, how do you say in English, rendimiento, like the performance, the yield has increased as well, right? So there might be some truth of it. We don't honestly know, right? So this is where like taxonomy and shapes and morphology comes into place. So the current naming system that we have for the cultivated cocas and for much of like the genus is based on morphological features. And in the case of cultivated cocas, pretty much rely on the leaves. So if you have a look, I guess that for Spanish speakers, understanding Latin is much easier. So if you compare the diagnosis of one of the varieties of cultivated cocas and of Erythrospermum coca novogranatense, you can actually see that some of the key features to differentiating them is the leaf shape, right? What you see here is outlines, some examples of outlines of these leaves. And so since then and now, we have been using actually leaves to differentiate, these different varieties. And for the police, it's even more relevant. As I said before, the United Nations Drug and Crime Office have been doing monitoring plantations, and they actually send personnel in the field to collect samples, herbarium material, which is then sent to botanists to identify specimens. And what you see here is a diagram that was done based on police information on the different varieties that they have informally named, right? You have like 15 or 20 varieties. And one key factor is, again, leaf shape, right? And this is, again, a non-trivial matter, because when you look at reports, like legislation, what you see here is scientific names underpinning everything, right? So we need to understand how much this names make sense? Do they really correlate with leaf shapes, genetics? We didn't know that. And this is where like our research comes into place. We have three simple questions, right? So can we really separate the coca varieties from the wild relatives? Looking at the leaf shapes. How do these leaf shapes link with names and genetic structures or genes? And something more of a nerdy question was, when these populations of cultivated cocas diverged in time, so how much human influence we had on the origin of these populations? These are like fundamental research questions, but I guess that my colleagues and I, like deep bottom, our dream with this research program is to generate like baseline knowledge to inform policies. Something that Jim said yesterday that like was really important is that as scientists, we have to do a stronger effort on like communicating the science we are doing to the general population. And this is like something we want to do. We want to create information that can help hopefully changing policies around the regulation of coca leaf and hopefully dissociate what coca is from cocaine, right? So it's perhaps a very ambitious dream, but we have to dream high, I suppose. So the modus operandi to answer these questions is very simple. My colleagues and I, we have been collecting shapes of leaves from herbarium specimens, digital light images from hundreds of them. We have been also sequencing these leaves. Two important things here on this program, this research we have been doing is that we have been very privileged in that we have access to material that was collected in zones of conflict. It's basically plantations where police, they go out and they shoot their way in to get there and do eradication programs and at the same time collect material. So the outcomes that I'm showing today won't include this. This is for future research. What I'm showing today is basically material that we got from our collection at Kew, which has, as you know, has a strong imperial background, but without that collection, we couldn't simply have done the research. As I said before, there is a strong regulation around the coca plant, and to a point where, for example, in the UK, the US, in several European countries, the coca leaf is considered in the same schedule as cocaine, which is crazy. Even in the Netherlands, for example, all coca species are regulated. You cannot simply just go and take leaves in the country because it's kind of the same as carrying cocaine. If you get caught, you can actually get into trouble, which is really nonsense, but what do I know? Another important aspect that we are actually trying to innovate here is to include type material from the wild relatives, something that previous research programs didn't do. And this is because I guess that whenever we use any material that is not the type specimen, it's simply an interpretation of a name that we are giving based on a description and a knowledge we have, right? So the only name to underpin shapes with DNA and specimen is using the type of specimen, right? And I guess that coca specimens is a blessed because they have so many leaves, and actually they are really sweet for extracting DNA. Where these old or new material, they are just wonderful. You get really good yields of data. So I guess that in an ideal world where like we make good decisions, we care about the planet, and unicorns exist, whenever you consider shapes or morphologies and you try to tease species apart, you get this kind of scenario where like these species come apart nicely. This is the typical iris data set, right? But what we found is very different. There is a lot to take in this plot. So what we did is that we took all these leaf outlines and we transformed them in mathematical coefficients using Fourier transformations. All of this was done by Rudy Diaz, who at that time was my master's student with Alex. He's a brilliant guy. He came up with calls and analysis, really good. This was done in cooperation with Dan Cheatwood, who is also based in the States. And what we found is that when you conduct a PCA of all the leaf outlines, you see a massive overlap of all these shapes. What you see here are examples of the different outlines of the wild relatives and the cultivated varieties, right? But what we are seeing here is that there is such amount of plasticity that you cannot really use names to differentiate the different leaves. So perhaps if there is one cluster that separates or segregates is Gracilipes, which is one of the wild relatives. But otherwise, it's really impossible to tease apart, the varieties by leaves, by the leaf shapes. Then when we ask a computer that is unsupervised clustering to try to decide how many different clusters you get 3-bit clusters, which they don't make much sense if you divide the samples, the number of leaves per specimens and clusters. So that's what these bar plots mean. For example, here you have a mix of Gracilipes, like different individuals from different populations. So we cannot really using like artificial intelligence intelligence tease apart the leaves by like discrete groups. You have three major groups. What does this mean? I don't know. Like, I don't think it's really meaningful. Like, short message is that we cannot really use leaves to distinguish the different varieties, the cultivar varieties, from their wild relatives. So the plot thickens. Then when we were like testing for specific characters that we know have been used to differentiate the varieties, for example, leaf roundness, we cannot really get a meaningful distinction between like the cultivar varieties and the wild relatives. This was done by Natalia Shiomska, who is now a lecturer in the University of Portsmouth. At that time, she was our postdoctoral researcher. But one very interesting thing we found is that when you look the ratio of basically what you have, if you have obate or ovobate leaves, which is a leaf inverted, right? What you see is a clear distinction here between the cultivar varieties and the wild relatives. And then Natalia came with a brilliant idea, which perhaps could be the first indication of a domestication biomarker. We cannot be sure, of course, but it's a nice hypothesis. And is that ovobate leaves in cultivar varieties could actually facilitate the harvesting. I don't know if you have ever listened to corridos prohibidos here. I see many people laughing, so you have. There is one song that is called El Raspachín de los Cocaleros. Has anyone heard that song? No? So basically the way coca leaves are, sorry? Actually, probably it is. It's in Apple Music. So, so basically, a raspachin is a person that goes and informally harvests the coca leaf. So it goes by taking the branches and then put the branch in between the finger and then just pulls the branch. That's raspar. That's like the way it's done industrially. Like in the indigenous communities, the harvest is only done by females and it's done in a very different way. But in industrial scales, like they pay like farmers, contract people, they just go and they scratch the leaves. So Natalia suggests we think that this could be perhaps an indication like for harvesting, but we don't really know, right? As Michael said yesterday, you know, whether these are real domestication biomarker or not, we have no clue. But it's an interesting hypothesis. So now let's move from morphologies to genes, right? So what we did is that, and just to clarify what we are doing here, what we did here is all builds on the data that was produced by Dawson and collaborators, plus our new sequencing. And then the reference that we are using here is these 400 local nuclear genes, which we use for all phylogenomic analysis and population genetic analysis. And what we see here is that when you conduct a PCA, you have a distinction here, a cluster between Novogranatense and Troxydense, so one like discrete population species. But then when you come to this side, you have kind of a merge between Gracilipes, one of the wild relatives, and the other cultivated species, and there are two varieties, right? And then you have somewhere here Cataractarum, which is the other wild relative of coca. We have highlighted here the position of the types. So that's an isotype of Gracilipes, and we have here the holotype of Cataractarum. So again, that actually shows the importance or the utility of sequencing type specimens whenever you can afford it, right? So it seems that maybe Novogranatense could be something that we could distinguish by genes or by SNPs, but then between Gracilipes and coca, we don't know. So that could reflect gene flow. We are still doing research on that. So this obviously tells us how similar these samples are in terms of like genes, but this doesn't tell us about like relatedness and ancestry. So we built a nuclear and plasmid phylogeny. Again, this was done using these 400 low copy nuclear genes, and this was done using entire plas genomes, I think. And what you can see here is that when you compare both topologies, you have a lot of gene flow, like basically nuclear and chloroplast 3 discordance, which could be an indicative of gene flow. What you can see here is that you have Novogranatense nested in Gracilipes, and then you have here some specimens of Erythroxylum coca. Here's the type of specimen of Gracilipes. You have the type of specimen of Cataractarum. So this is a very similar topology to the one that Dawson retrieved. But then what they didn't do research on is how does this look from the chloroplast side? Then you have a very similar, very distinctive topology, as I said before. You have here 2 specimens of coca. There is another one here. There is Novogranatense, which retains, well, is recovered as a clade here we could support. So whether the scenario of 3 independent domestication events is true, we don't know. I think that we need more sampling and more research, right? I think that there is a strong signature of hybridization here to avoid, that prevents us to really say that we have 3 independent domestication events. And then the last thing that we wanted to understand is, as I said before, when in time these populations diverged. So something that we did that it was in collaboration with Gustavo Allen, who at the time was at the group of Asif and Yan in UCL, ULC, sorry, is that we wanted to first understand or to try to trace the ancestry of the genomes of the cultivated cocas and the wild relatives using a mixture analysis. And when you assume 4 or 5 populations, ancestral populations, what we see here is that Erythroxylum coca, variety coca, retains or kind of makes a distinctive population, and so granatense. But then one of the varieties of coca, which is variety paru, comes or is its ancestry, the genome ancestry is retraced to Gracilipes, right? So using a coalescent approach in BEAST to date this tree, instead of dating every individual as an independent terminal, we managed to find out that actually the divergence of this population from Gracilipes and Ogratense from Cataractarum happened or preceded by much longer the arrival of humans in South America 15,000 years ago. So, which might suggest that these populations were actually there since a very long time. But then what we don't know really is when in time these individuals started to synthesize or produce cocaine. That's a matter of research as well. If you want to read more, here's our publication we came out this year. This is what is published now. The next steps, what we are working on is on assembling a genome of high quality. We managed, using only nanopore sequencing data, to retrieve a genome which is, has a scaffold, 300 scaffolds, but the level of completeness is quite high. There was a genome published last year in PNAS, which actually unraveled the biosynthesis of cocaine using synthetic analysis of tropane alkaloids. So our plan is to assemble or to do a guided assembly of this reference genome of our fragmented genome to this reference genome. And then we are analyzing a bunch of accessions for biochemistry. So that's the slide I showed at the beginning. We have about 100 samples that we have analyzed from our collection using GC and MS. And we're also sequencing all these specimens that were collected in zones of conflict. So we don't have enough hands, and we are actually looking for cooperations if anyone is interested. The nanopore data could be very, very useful to learn about methylation patterns and gene expression around the genes that are responsible for cocaine synthesis, right? So if anyone is interested in cooperating, just, we would be very happy to work as a team. So just to finish, and this was a very short talk, short and sweet. I hope I convinced you that coca leaf just goes beyond, what we know now, all the violence and conflict. It's a plant that has, a very, very long-standing history that is relevant today. That basically, coca doesn't equal cocaine, right? It goes well beyond the drug party. Based on the results we have gotten, I think that an updated classification is warranted, provided that we have more sampling. This reminds me of the discussion that happened yesterday about the morphological plasticity, where we should lump or split species. Some colleagues actually would like, based on a phylogeny like this, to split every population here as an independent species. But would that make sense? I mean, considering all the legislation and information that there is around these names that we have been managing for years, that would create havoc, right? So we have to really be careful before we make, important decisions. That's it. I hope that you enjoyed the presentation, and yeah, happy to take any questions. Oh, before I forget, this work is only possible thanks to all the cooperation that we have established. In particular, I'd like to thank Alex, who has always been keen on supporting with ideas and funding our crazy ideas. We went to Foundation and they were like, "What, are you going to work on coca trees? Are you crazy?" So, but Alex has been always very keen on that, lending us his mind and financial muscle to develop this. So, yeah.
Hola, ya, ahora sí. Muchas gracias, Oscar, por esta presentación, este seminario que yo creo que va a traernos muchas preguntas ahorita. Aprovecho ahorita para decirles que Oscar nos va a dar un taller el próximo lunes sobre secuenciación Oxford Nanopore, también parte de estos eventos de celebración del 95 aniversario. Entonces, dejo abierto para las preguntas.
That was great, Oscar. And I actually wanted the word coca like 15 years ago. So that's
Yeah.
you population sampling of the different variety groups. So you have and I forget now. You have whole genome sequencing data for them, or
So when we conducted this analysis, we used as a reference 400 low copy nuclear genes, but now we have a reference genome, which is fragmented, but then there was another published. So we're actually rerunning the analysis with the whole genome.
so it would be really interesting to do the population genome,
Yes.
right? So I think that well, we can talk, or you can talk to Rafael as well.
Yeah. Thank you.
Thank you for the talk. I like the genus. We did a lot of work with one erythroxylum in the Chamela field station, Erythroxylum habanense, that is heterostylus. And I think most of the genus is heterostylus.
Yeah.
So, well, you have not mentioned that. I guess that there are no flowers or few flowers usually to do the analysis. But the thing I want to commend you is that if you are going to do high quality genomes, would be very nice to try also to find out the genetic basis of the heterostyli.
Yeah.
So, well, but maybe you can comment something about the reproductive biology and also the dispersal. They are bird dispersed, at least in Chamela. And in Chamela, the trees flower in a very synchronous way after few rains. So I think there is a lot of interesting natural history around that.
Yeah.
Maybe it's very difficult to study in this species, but maybe it's interesting.
Well, like many domesticated plants, it's really hard to find cultivated cocas in their native environments. What remains is what you find in these massive plantations or like in indigenous chacras. Yes, the cultivated cocas are heterostylus as well. I didn't talk about that because, again, as you see on the diagnosis, like only on this diagnosis here, actually, is mentioned like the flower and like the corolla that has some spikes, but here, there is no reference to it. So it seems like the flower is not really, an important character on teasing apart the species here. When you go to plantations, the coca plantations are not really led to be flowered. Indigenous populations and plantations, they do. And actually, they exchange seeds. All the plantations, most of the plantations, the illegal plantations are clonally propagated. So flowers, there is like, you basically are working with unfertile specimens. So it's really tricky.
¿Alguna otra pregunta?
Hi, nice talk. Have you used leaf architecture and see some difference in leaf architecture in the varieties, or no, only the leaf shape?
No, so far we have only looked at the leaf shape.
Well, maybe.
But that's something that could be considered, yeah.
Okay, thank you.
So thank you so much for the talk. It was really, really good. And I was wondering, because, we have similar things in Mexico, that we have some plants that are used as a drug or something. So I totally agree with you and the influence of scientific in the public policies. So my question was like more related with what will you say it is needed for the public policies, either in Colombia and the UK? I mean, you are working in the UK, obviously they have some kind of different regulations. So with all this work, what would be like the main, the main thing that you want to influence in those policies?
I guess that when I look at the regulations, at least of the UK, which I'm familiar the most, is that there is like lack of knowledge on the tradition and history of the coca leaf. And this is because there is a lack of baseline knowledge on the attributes of the coca leaf, right? There has been some research about the medicinal values, the properties, but it's not stuff that we are doing frequently. There is a lot of interest on this biosynthesis of cocaine, of course, which is used as a medicine in some countries. But I guess as researchers, if we promote maybe a research program around, these knowledge gaps, then things perhaps can change. And if we actually work with the government and we form the government and say, look, these are our findings, I think that these kind of things are actually more easily done in countries where I live now, like the UK, because there is a more stronger connectivity or connection with scientists and the government. then things perhaps can change. When I was doing my research, I actually found out that in 2012, there was an attempt to change the policy in the UK. There was a group that was trying to lobby to say, look, the coca leaf, you know, it's not as evil that you cannot synthesize cocaine if you bring dried leaves here. But, you know, the government that was in power is very narrow-minded and conservative. So, but more science is needed, basically.
Oscar, it was a really awesome talk. Thank you. I was wondering something about your methods for geometric morphometrics. Normally, people do the PCA with landmark-based morphometrics, but you and Dan Chitwood decided to go for Fourier harmonics. And I was wondering, like, why to do Fourier harmonics versus landmark-based morphometrics?
That was a suggestion by Dan Chitwood, I have to confess. So he was guiding us and Rudy on this. So I cannot really tell you why, but you know, we trusted Dan Chitwood, who has obviously much more expertise on this than us. So I think that they are actually revisiting this paper, but with what they call a garden experiment. They have access to leaf data from a farm in Colombia that has is the largest police farm with coca plants that they have found. And I think they're using different method. And when you compare the results with what we have found, they are very similar. But yeah, I think that for that, we have to ask Dan. So he's the expert.
So I have enjoyed coca tea, and it would be nice for people to understand that we can get coca tea and it's not illegal, right? Was its use for making tea, was that the original use by people in the Andes? And how diverse are the species or varieties that folks use for making tea?
That's so about the use. The traditional, I think that they make the use of coca leaves to make tea something more new. As far as we know, based on, again, the rich archaeological record, is that coca leaves have mostly been chewed. If I remember correctly, the oldest archaeobotanical record we have is actually dried, chewed coca leaves. And again, there's always the association with this calcareous powder, either drawn from seashells or from actually our plants, like jarumos, Cecropiaceae. So this thing of doing teas and stuff is something more like recent. There is another alternative practice which doesn't involve the chewing of the leaf, but the preparation of what we call harina de coca. It's like dried powderized leaves and then mixed with, again, another calcareous powder, and then putting that powder in your mouth. So you can actually buy that in Colombia. So the tea is more recent. Now, the question that you're asking about the different varieties, that's a very interesting one. Something that we want to figure out is where there is any genetic structure on indigenous, plantations or varieties. Like, for them, it's really important. So it's way more than a plant, and they have their own names for the different varieties they consume. Some of them are being called cocas dulces in their languages. At a point, it's so important for them that when they meet each other, and I got to see this in person when I did field work in Sierra Nevada last year, when two indigenous people they meet, they actually don't shake hands. They exchange coca leaves, right? So they have their preference for the leaves. So whether these, varieties reflect on genetic structures and different populations, we still don't know, because there hasn't been much sampling on these areas. It's difficult to work there as well.
Hola, perdón, sí, cuando estuve hay regiones en los Andes de que toda la comunidad, excepto los niños, traían en su boca, como si trajeras chicle, así, y te hablaban, pero cuando les pregunté por qué razón, y decían, porque trabajamos todo el día en el campo y no tenemos tanta hambre por tener eso.
Yeah.
Pero sí me fijé que me dijeron, pero a los niños no les permitimos traerlo en la boca.
Yeah.
Y la otra comentario es, ¿por qué no usan el término cultivar? ¿Por qué variedades en plantas cultivadas?
So to translate for everyone, our colleague is telling of a story of that was in Peru, ¿cierto? ¿O dónde fue? En los Andes. So somewhere in the Andes, there's a community of people carrying a pellet of leaves in their cheek, which is exactly what you see here. That's the way, one of the ways of consuming coca leaves. There is a very nice book by Goltimer, I think. It's very, very old, about the history of coca and the Incas. And one of the relates in that book, which is really cool, is that when the Spaniards came and they were just killing everyone, destroying everything, they tried to take out the coca leaf from the population. And what they were saying is that basically the indigenous people were not working anymore. Like they couldn't sustain long journeys, long working hours. And this is because the coca is a heavy stimulant. It doesn't get you high or make you crazy, but basically it takes, the hunger, the thirst. It's a stimulant. It's a very good one. It's just like a good cup of coffee, but maybe more concentrated one. And then regarding the question about why do we use varieties and not cultivars, this taxonomy classification that we have been using for the last 40, 50 years comes from Timothy Plowman, who was a very, very good botanist, US American, who sadly died at a young age. And he basically named us varieties. What is a variety? I honestly don't know. That's very subjective, right? So.
Sí, y a propósito de cuando subía más de 6,000 metros, me daban té de coca.
Yeah, it works.
Y sí.
For what we call el mal de Soroche, yeah, it helps.
Sí.
Yeah. So.
Bueno, gracias.
Hi. Thank you for the talk. It was great. What about other cultures that you say is a pantropical plant? So what about people in Malaysia, Sumatra, don't have ever used coca?
As far as we know, no. So there is not much literature about alternative uses for different species beyond like the nootropics. As far as we know, it's just these two varieties. So because of the ones we have analyzed, or like our colleagues have analyzed, in terms of like metabolites, only the two cultivated varieties are test positive for like cocaine or tropane alkaloids. So, but I suppose we haven't analyzed all the 270 species, right? So who knows? But there is so much we don't really know, and yet we demonize everything.
Thanks.
Hola, muchas gracias por la plática. ¿Sabes si, no recuerdo si lo mencionaste, si hay algunos datos de ciertas preferencias por las culturas, por alguna de las variedades, y que esto esté relacionado con la distribución y con el tiempo de origen y con el grado de diferenciación entre ellas?
That's what we are trying to answer. I mean, for sure, there is a preference for certain varieties in some populations, indigenous populations, like some of them actually like less bitter cocas. That's what I was saying before about cocas dulces. So there is for sure a preference for certain traits, like organoleptic traits. But where this has left a signature, like in the genomes, of there is like any sort of population structure, we don't know, because the sampling that has been done so far, not only by us, but by different colleagues, the individuals that come from indigenous populations, like plantations, is very minimal. So we don't know.
¿Alguna otra pregunta? Seguro. Sí hay, yo sabía.
Hola, Oscar. Gracias por tu plática, me encantó. ¿Has considerado analizar la anatomía de la hoja? Porque, bueno, la morfología, pues, está muy correlacionada con cuestiones ambientales también, ¿no? Pero la anatomía a veces te sorprende con caracteres que te pueden ayudar a explicar todo esto.
That's a great idea. Something that we have been thinking about is, so we looked at the leaf outlines, right? But what about the venation patterns? That's something that we could look into. That is, as I said before, it is really so much work, and this is like, this started like as a side project for us, and it just grew. So we're actually trying to pursue more funding and like get, more hands-on. But if we ever get there, which I hope we do, then for sure we will do that. So looking, for example, at venation patterns, using artificial intelligence to see if you have like clusterings, that would be great. Yeah. Thanks for the idea.
¿Punta? ¿Alguien más a la 1, a las 2 y a las 3? Muy bien. Bueno, ahora no se muevan de sus lugares. Lo que queremos hacer ahorita es, si Oscar Bueno, le damos las gracias, le damos un aplauso por su seminario. Eh, Oscar, si nos acompañas aquí adelante en el público, vamos a tomar una fotografía de todos para que tengamos el registro. Sí, solo tomar asiento por aquí. Ajá. Y la voy a tomar con mi celular, pero tal vez alguien me quiera dar un celular de mejor.
Tenemos una foto que pues puede coger con la neta. If you guys could stand up over here.
¿Te pones en un lugar? Es que solo tú no sales. Listo. Ahora, como el día de ayer los invitamos a tomar el lunch junto con nuestros invitados, entonces tráiganse su comida, su torta. Yo voy por la mía ahorita aquí al Instituto Vecino y nos vemos en las mesitas aquí en el Jardín de la Espiga. Un segundito, les vamos a entregar las constancias a los ponentes, ya se me estaba yendo. En un segundito la doctora Susana va a tomar la palabra para hacer esta entrega. Si nos dan 2 minutitos. No me sé ningún chiste, así que vamos a tener que esperar. Sí. Ah, perfecto. La pluma apenas ya. Ah, no es el puntero. Ya. Listo. Tenemos.
Well, the first one, we can ask Mike Puruganan to come here, please.
For Emily. One is for Andrea. Por Socorro González. Listo. Ahora sí a comer.